Hello, good afternoon, everyone. A few days ago, I think I have shared with you a paper that is titled Induction of Pluripotent Stem Cells from Adult, a paper that is a previous one. Okay. I shared a paper titled this one, Generation of Transgene-Free Induced Pluripotent Mouse Stem Cells by Piggyback Transposon. Right? And I guess we haven't discussed that before. So today we are going to discuss this paper. In the last unit, we have 4 papers, I think. In that, this is one of that paper from 4th unit. So as a part of your assignments too, you're going to discuss about 2 papers. Taking now is also to help you or what I am looking for. Okay, so let's get going. So quickly we'll go through some background on the paper. So this paper first has some things like induced pluripotent. That is one technical term. We need to know what is meant by transgene free, right? And we need to know how can piggyback transposon help in doing that. Okay, so the background will explain you some parts of the title, and also there is stem cells. We should have known these things, otherwise the title is completely new, or we don't understand it properly. That is why speaking about the background is important. What we are trying to do in regenerative medicine, in the first introductory class that we have ever discussed, how rDNA technology and stem cell technology can come together to describe. One second. Yeah, am I audible? Is the network fine? I thought there was a disconnection in between. Was there a disconnection?
No, sir, it's fine. No, sir.
Okay, okay. Yes, that's fine. Good. So in the first topic, we are trying to talk about how we can repair, replenish, or regenerate lost or diseased organs. In that case, we should talk about, we already spoke about, we had an introductory class on epigenetics. A video was uploaded. So the central part of it is about generating patient specific. When you say patient specific, that means what the tissue that we are putting into the patient should be compatible with the patient, right? We cannot, if we take from some other person and if the HLA does not match, then the tissue implanted is rejected by the patient's immune system. So the idea is to take cells from the patient and generate, then induce pluripotency. The differentiated cells, the patients that were taken, something like from skin biopsy, for example, are completely differentiated, right? And we need to generate induced pluripotent cells. When you say there are multiple types of potency in a way, you call it as totipotency or pluripotency or multipotency and unipotency. That is, if one cell can give rise to multiple tissues and then ultimately give rise to one individual or an animal, viable animal, then you call it as totipotent. If a cell can give rise to only all types of cells, all but not the complete organism, then you have, they are called as pluripotent stem cells. In the future, medicine is going to be something like this. It could be. If an individual is there, they might take some cells of that person and say they are differentiated ones. They cannot give rise to any other cell types. They could be reprogrammed to pluripotent stem cells, and these will be stored in a stem cell bank. If this individual anytime in the future has an issue, they would take the iPSCs from the cell bank, they would derive that particular differentiated cells, and then they will put it to the into the individual. That is likely to happen. Already there are indications, there are stem cell banks already in our majority of the cities, where they store the umbilical cord blood stem cells. But this is going to happen in the near future. So I hope you understood what is meant by pluripotent and why we want pluripotent stem cells. The embryonic stem cells are the pluripotent ones. But this individual is already grown, and there is no way of getting embryonic stem cells that are of his haplotype directly. So we can induce a cell. That is, if you take a differentiated cell and you induce some by the, or give the ability of that cell to give rise to different types of cells, all types of cells, that means that is pluripotency. And you are inducing it artificially. That's why these are called as induced pluripotent stem cells. And then you can proceed to either two different ways. You can try to generate these cell types that you want for a damaged tissue, and then you can give those to the patient. If the patient had a genetic defect, then one can try to repair these induced pluripotent stem cells, something like gene therapy. We have learned in transgenic animals the same way one can make it ex vivo gene therapy. We will have a lecture on that some other time. So it's kind of gene therapy, and then you make healthy cells and then put it into the body. So this is the central view, or what is going to become the future of medicine. That is, regenerative medicine is going to revolve in this way. So I hope you understood what is pluripotency and why do we want pluripotency. Try to learn a little bit, or try to get familiarized with yourselves with the terminology, differentiation and de-differentiation. If you have a pluripotent stem cell and you're making it into a somatic cell, right? You're deriving a somatic cell from a pluripotent stem cell, that is called as differentiation. If you are having a differentiated cell, and you're making iPSCs from here to this end, that is de-differentiation. In the way we are trying to change this, I hope you also realize that the genome of the pluripotent cell and also the somatic cell is absolutely same. It's like in different cells in our own body, the genome is same, but then there are the morphological differences, or in the cell at the cellular level. That is because of the majority of it is because of the epigenetic modifications, that is the nuclear program. So here we are trying to reprogram the nuclear DNA so that the cell attains different form and functions, and that is between differentiation and de-differentiation. In all the cases, you can think of it as one cell has a kind of nuclear program, something like pluripotent cell nuclear program. And if you are changing it to a somatic cell, then you are changing the nuclear reprogram. You're changing the nuclear program. Okay, so I hope you understood that part. And I want you to remember those clearly so that you'll not be lost anywhere during the course of this class or until we finish this paper. So some time ago, or in 2007, a decade almost ago, there was a paper, Induction of Pluripotent Stem Cells from Adult Human Fibroblasts by Defined Factors. So in this work, and there are many other that they determine the minimal set of genes required to convert a differentiated cell. Okay, I will write the differentiated cell, I'll draw it like this, into a pluripotent stem cell, something like this. So this is pluripotent, this is differentiated. If we want to de-differentiate a terminally differentiated cell into a pluripotent stem cell, you need a minimal set of 4 factors. Those are OSKM. We will see them. OSKM, OCT4, SOX2, KLF, and CMYC. These are also referred to as Yamanaka factors because this pioneer has he has pioneered several majority of the research or did significant contributions in that, and he has received Nobel Prize in 2012 for this, for developing this technology. So I don't need that you have to memorize all the functions of these, but what you should remember is that this OCT4, KLF4, SOX2, and CMYC, they are transcriptional regulators. They regulate many other genes, and eventually they regulate the expression of many other regulators. And then you can see that that these 4 factors have huge potential for the ability to reprogram the cell. So remember these factors: OCT4, SOX2, KLF4, CMYC, OSKM. Just remember that way. So as I was telling you about these factors are transcriptional regulators. They regulate several target genes, which themselves are transcriptional factors, or they can be epigenetic modifiers. So as I was telling about reprogramming, you want the nuclear program of the somatic cell or of the differentiated cell to become undifferentiated or stem cell. It's actually pluripotent, right? Pluripotent stem cells, not adult stem cells are also there. So if you want to do that, you need epigenetic changes that should be made. So you will need changes in, or the expression of different transcriptional factors and also epigenetic factors so that the nuclear program can be changed. And the cell that was displaying a morphology like this would become something like this. So if you want to reprogram, you want to make a genetic reprogramming of one cell to other, you now need, we know what are important. OKSM, you can write OSKM either way. These are the factors that are there. And assume that we also have the cells. Now, what we are supposed to is to make up and make pluripotent stem cells. That means the genes responsible for these proteins or factors should somehow be incorporated into the cell and expressed so that you will be able to generate induced pluripotent stem cells. So we obviously would need genetic factors using, they can use viral vectors or they can use transposon based vectors. I guess you can recollect when we read about the vectors, we did learn many details about those things. Here, and there are also non-genetic ways, but they are kind of safe, but they are not very efficient. The genetic ones are very efficient because when you deal with the gene, a gene can produce many mRNAs, and each of those mRNAs can produce many proteins. That makes it much more effective. Whereas if you're trying to give directly the genes that are already made, proteins that are already synthesized, there is a limit on how many proteins are being made. So they're not as efficient as genetic vectors. But the genetic vectors do have a problem because if we are using genetic factors and they may get integrated into the genome. I hope you remember a kind of statement I used in when we were discussing about transgenic animals. I would say it should have it is something like this. You have three genes, or so say you have X gene here and Y gene, and you're putting gene A in the middle. When you're talking in a when you're defining what you want in transgenic animal, I would have to say a statement like this: I want gene A to be incorporated between X and Y only, and I mean nowhere else. Right? That is high definition. And also you should say, I have to make it a more clearer sentence that I want integration of only gene A between X and Y and nowhere else. That is highly defined, right? That means why do we need such kind of things is because anytime we make any integrations, and if there are any unintended integrations or inadvertent genetic modifications, then they are going to we are not going to keep track of those things, and they might result in complications. Say for example, they might become carcinogenic, especially if it is something to do with the way we deal in terms of clinical ways. Then we want to treat the patient. We don't want to add a disease to the already existing diseases in the patient. So one has to exercise a lot of clarity. We have one assignment, isn't it, where one of the assignment which I said is compulsory is about potential limitations, risks, and ethics in using rDNA technology. So I think I want you to think of all the possible issues that might come up, and hopefully you will be able to remember those for a long time. Anyway, the point is there is always a problem of there is integration, there is a problem with the safety. Okay, but some of the genetic which may not be integration free. That means you're giving it as an episome. I hope you remember what is episome. It is free of content. We rid of those episomes or set those cells that are free of the episome and then use it for your purpose. We'll define use of this paper. Can you hear me? Can somebody come?
Sir, your voice is breaking, sir.
Vasudha, can you hear me? Okay, how is it now? I just lost the connection.
Yes, sir, we can hear you now.
Is it all right
Yes,
now?
sir.
Hello?
Yes, sir.
Yes, sir, it's fine
Okay,
now, sir.
thanks. Yes, I lost the
It's
connection.
working, sir.
Okay, we are back. So let me describe the objectives Okay. So the first is generate a transgene-free induced pluripotent stem cell. I hope I tried to explain you some of it until now, so that you will be able to understand the kind of what's used and so on. Transgene-free, which means we don't want any kind of change in it, and we want to generate pluripotent cells, iPSCs. So we have to discuss about the methodology of thing. When you are doing your assignments, I would want that you read the methods very well. Methods, if there is such article you haven't read too light in terms of abstract going to figures. um, can you hear me? Can you hear me?
Yes, sir, we are able to hear you.
Okay,
Yes, sir.
sorry again. Yes. So if you did not read the methods and methodology of a paper, you haven't read the paper. That's the principle you have to remember. Without knowing the what methods they have used, we cannot come to any conclusions. So don't take it for granted. You should always exercise healthy skepticism, irrespective of who said and what they said and where it is published. It doesn't matter. Okay, so first thing is we will understand their methodology. Then we will look into characterization, how they characterize if pluripotency has been induced or not, and then the evaluation to show that they are pluripotent. So these are the objectives of this paper, and we will try to look into that. As I was telling you before, there are retroviral or entoviral genetic ways of inducing pluripotency. It is something like you take a lentivirus, for example, put in OCT4 in one, and then they make a phage out of it. Okay, and like that you can generate different phages, each containing one gene, and then infect fibroblast or some other differentiated cells and ensure upon growth, upon expression of the OCT4 OKSM factors or Yamanaka factors, the cell over a period of days will change into pluripotent stem cells. Okay. The problems with this method is that it is genomic integration. We don't want that. There are risks with it because they might form tumors, and it is never safe to have unintended integrations in it, or we don't want any kind of integrated fragments into our genome. You can also use viral transduction. Some people have suggested, and in this case it is integration free in the sense it will survive as a separate entity, like an episome, and then maybe we can select transgene free ones. There is, because of which there is no integration, and there are, because no integration, unlikely that they would form any tumors in the chimera. But the efficiency, I think, is not as good as in the case of retroviral ones. So similarly, you will also have plasmid type or purely episome type of episome type of vectors. You can generate iPSCs. The problem is that the efficiency is not very high, and we don't know if any part of these get integrated anywhere. We are not able to track those. That is one of the problems with them. So ideally, any induced pluripotent stem cells that we are planning to use it for clinical purposes in the future, The technology should not have any, the cells should not have any genetic changes in the genome. And they should have appropriate expression of OKSM factors, because only they are capable of reprogramming differentiated cells into induced pluripotent stem cells. So that means the reprogramming should have happened. So here we are going to, now we are going to read or try to understand a few techniques or components these people have used. And then we will try to go through the paper. Here they have used piggyback transposons. I hope you know about transposons, right? I want an empty slide. Okay, I would use it here. So I hope you know about transposons. In the last class also, we discussed a few things about generating transposon-based mutant library. So transposons are, you can have copy paste or cut paste. They have the inverted repeats at the end, which are essential to be recognized by the transposase enzyme. It recognizes here and here. Say if it is cut paste, then it would have cut the DNA somewhere, the transposon like this, and it would choose a target DNA. Say for example, I have a sequence something like this. I have a sequence like this. So the complementary is something like this. Okay. Now, if the transposon is moving in, the transposase enzyme will make a cut. The cut is likely to be somewhere here. Say, for example, not all of them are sequence specific. These are usually, they are not sequence specific. Sometimes some of the transposons are integrate into high AT rich regions and so on. There is a little bit bias, but it depends on the transposon. So if the transposase cuts the target DNA in the way I have shown, then you would have sequence would have been split into CGA, then you will have CGT, GTAC, and the remainder of it I'm writing here, CATG, AC, and TG. Okay. Now this transposon that is present here, this transposon is going to get integrated here. So I'm going to draw it again, just representing the borders. So these are the inverted repeats that are essential to be recognized by the transposon and for mobility from one place or transposition. Now we have some spaces. This is a space, right? And so is also this space. The DNA repair mechanism can repair it, and it will include the DNA. It will make C, A, T, G, and here it will have G, T, A, C. I hope you can notice that in the beginning, the target sequence actually has CATG once. Now, if you look at after transposition at the target site, what happened is CATG is one sequence here, and again you see the same one here. You see duplication, and that duplication is present at the target site. So you call it as target site duplication or TST. Imagine now this transposon that is present here is moving out. Cut paste method, assume that. What would it then have? What would the sequence then be? It will be GCA CATG. I wrote this sequence, and now I'm going to write this, the other part. That will be CATG AC. The complementary strand CGTACTG. Okay, I hope you can notice that this is the sequence, duplicated one. The original one had one CATG, but now you have two sequences of CATG. This is the duplication. So majority of the transposons, they cause target site duplications like this. So if we were to use any normal transposon, and the idea, that general idea is that if this is the transposon, say for example, I want to have the transposon with the inverted repeats, and then I want to have the 4 genes O, S, K, M, right? I can hopefully from the vector, I can integrate it into the genome, okay, so that the transposon is integrated. After reprogramming, I can try to remove this transposon. Now, if I use such a kind of strategy, what is going to happen to the target site? TSD is going to happen, that is target site duplication. That's why the ordinary transposons that we know of are not very useful, because even at the smallest scale, at this level, they are going to make a change in the target site, which might be drastic sometimes and cause problems with the cell. I want to also remind you about, I used a term or a phrase in the last class, wings clipped element. What does that mean? I noticed that in this particular transposon, we did not include the gene for transposase. Transposase would have been somewhere else without any integrated repeats. This is the gene for transposase. It will produce transposase, and that transposase will mediate the transposition. Okay, we did not, because we don't want this artificial transposon to move from one place to another place. So we have to separate the inverted repeats from that of the transposase gene. Otherwise, together, they can jump from one place to another place. Separated, they cannot jump. Then we have much more control. That's why transposons do seem to, they do appear to have potential for use in recombinant DNA technology, especially in this case. But there is one limitation, that limitation is target site duplication. So for that, what we have is, there is a nice transposon that's called as piggyback transposon, and they are fantastic because they do not leave a target site. There is target site duplication, but when they are leaving out of that place, when they are cutting out of that place, they make the sequence as they restore the sequence to its original state. We will see that now. So this is the transposon. It has this from here to here. You can assume that's the kind of transposon it is. And usually you have this is the target site. It's already integrated. And now we are trying to see if it is going into a new target. Okay, I mean, from here, what will happen? The piggyback transposon has a high affinity for sequence like this, TTAA. You can see TTAA, and here is TTAA. The transposase enzyme makes a cut here and also a cut here, and it forms a hairpin loop like this. I hope if you read Watson lesson, they have a there is a nice picture in it, how the first strand breaks cut and the second strand break cuts, and how it is transferred from one location to the another location. There are differences. I would like you to watch those things. Look up in the textbook, and you can understand better. So the the hairpin that is formed like this is now going into a new integration site. In the new integration site, it is going to make a cut like this here and here. Okay, so or probably I should do the reverse of it. In the new integration site, it is going to it's going to make a cut, yeah, sorry, here and here. So it is going to make a cut like this and separate. So the lower strand is here and the top one is in here. Okay, and the transposon that was in this location with the hairpin loop is now going to come and integrate itself here. And when it is integrating, it will make a cut in this location and in this location within the hairpin. The hairpin should be broken, otherwise it cannot integrate into the target site. When it integrates, now you have, it has preference for TTAA. It is TTAA, right? And so is TTAA. I am reading it in 5' to 3' direction. And now it already has a sequence TTAA. These two are complementary sequences, and it will base pair here like this. And the same thing also happens on the second side. And now the repair, upon repair, you will get complete double-stranded DNA. Look at it again. I will erase these so that you will be able to see. What is the actual sequence? The sequence is TTAA. What is in the after transposition? What do you see? You have TTAA and TTAA. There is target site duplication, right? But then let's see what is happening to the target site that was from where the transposon has just come. So the transposon has this is the transposon and this is the target site, TTAA, target site duplication. Upon cutting, what happens to this part and the second part of the target? Because this overhang is complementary to the other overhang, they will just fuse up and will form TTAA. There is no target site duplication. Now there is no footprint that is left behind, isn't it? The original state of this one would have been TTAA, something like this. Upon transposition, it became duplicated. Upon excision and moving, cut and pasted somewhere else, you are restored to its original state. So I described to you in the previous slide how target site duplication may happen. And with the piggyback transposons, I hope you can see that there is, after excision of the transposon, you do not see any kind of target site duplication. So this is a good candidate vector for transgene-free induction of pluripotency. Like transgene-free genetic genetic method for integration iPSCs, for generation of iPSCs. So you have the same picture. I don't need to describe that, I guess. So there is some more theory that we need to know. What are the points we should know about how to generate multiple proteins from a monocistronic mRNA? There are problems, right? I gave you, this is one of the questions in the assignments. I think, how can one mRNA generate multiple proteins? That's the question. As opposed to prokaryotes and the eukaryotes, there are differences in the way mRNA is. mRNA in prokaryotes is polycistronic. In eukaryotes, it is monocistronic. Typically, that is the case. Monocistronic Can you hear me?
Yes, sir.
Okay, thanks. Sorry again for the interruption. So I was talking about polycistronic and monocistronic. In your majority of your videos, you have given an explanation like this. The gene will give rise to transcription, and there are in eukaryotes you have introns and exons, isn't it? So, and there is, so the open reading frame will have to say remove the introns and generate an exon. And this mRNA can be, the mature mRNA may have 1, 2, 3 exons or 1, 3, 4 exons. So you are generating two different types. But from one mRNA, you are only getting one type of protein. From this one mRNA, you're going to get only one type of mRNA, protein, right? So now I want to, if I want to generate, say for example, if I want to generate a situation where I have 4 different factors, I have 4 genes, O, K, S, M. If I need to express them, I will have to construct something like this gene of OK OCT4 with its promoter. Then you need a terminator, then again a promoter, K, terminator, and so on. S, you have to have promoter and terminator, and like this, OKSM. Right? The genetic element is too huge, and the promoter elements sometimes do occupy a large amount of space, genetic space. You are working with artificial mechanisms, and we would want to work with smaller ones. And smaller the size, less possibility of unintended recombinations. That is one of the problems also that will be there. So how can I reduce it? What if I have, if there is one mRNA and I have one promoter and these genes are present something like this, and there is one terminator. So you'll have only one, there is only one mRNA produced, and at the end you get 4 different proteins. Then you are drastically reducing the amount of genetic space. You can do it in one vector itself. Otherwise, typically some people do other approaches, one vector in this, another vector with K, another vector with S, and so on. That is complicated stuff. In eukaryotic biology, you do have, sorry, certain, I think it's mostly associated with viral proteins, where they have one mRNA, and the mRNA has, it can produce, it is a long protein, long mRNA with 2 different ORFs. Okay, there are 2 different ORFs. So as you can recollect, the G cap is essential, kind of essential for transcriptional initiation. EIF4 binds to it, and then pre-initiation complex is organized, and then the initiation happens of translation. But if the second ORF is here, how can the ribosome initiate or arrange, organize the initiation here? That is one of the questions. In that situation, there are something called as IRES, internal ribosome entry site, right? Internal ribosome entry site. So they are kind of small sequences onto which ribosomes can make initiation complex and then translate the second one. So from one mRNA, you can generate 2 different proteins. But what you need is having the IRES. Okay, that is one possibility. There is another also. I think it is with the majority of the cases. It is with the viral parts. There is one mRNA, and it has one single ORF. The only point, however, is that upon translation, it produces a big protein, but that protein gets broken down into 2 different proteins. Okay, so from one mRNA, you can get 2 different proteins. It is translated as one and produced as, and ultimately you have 2 functional proteins. That's nice. So why I specifically like this paper is they have used these kinds of technologies or strategies. They identified the best of the solutions. You have a goal to generate transgene free. It should be smart. It should be error free, and it should be the smart way of doing stuff. So these are called as the place where they break, right? That is, there are small peptides, something like 2A peptide. These are called as self-cleaving peptides. Self-cleaving peptides. So there are multiple different types of peptides that are identified from different viruses. And viruses produce one huge protein. The same also I think is with the coronavirus too, COVID as well. So they produce one large peptide, a large protein, and then they break it into smaller pieces, and then each of them is a functional protein. So they use these self-cleaving peptides. The design is something like this. I want to have OKSM or OSKM, whatever be the case. They generated a whole one single open reading frame. Starting with a start codon would have been there with O. Then there is a small peptide, like the self-cleaving peptide, like indicated here. And then they have added another gene, small self-cleaving peptide, another gene, and so on. Upon transcription and translation, you would generate a huge protein. And because these peptides are self-cleaving, they would cut themselves up here like this. And thereby you are generating 4 different proteins. There could be a problem in the sense, now the protein, okay, OCT4 is what you want. But now what you have is OCT4 with a small P2A tag. If this tag interferes with the functions of OCT4, then this is not functional. So if we want to use such kinds of self-cleaving peptides, we need to check or ensure that the peptide that is going to be now translationally kind of fused with the OCT4 does not interfere with the function of the OCT4. I am assuming in the paper also they have used 3 different peptides. Why do you need 3 different peptides? Why not just the same self-cleaving peptide everywhere? I'm assuming again that the probability of these peptides interfering in the function of the protein might have played a role in the others choosing different types of peptides in different locations. Okay, that is one way of seeing it. I will just go through the rough sketch, and tomorrow in the next class I will repeat from the previous slide. Okay, so they have generated a huge vector kind of, and now you can see that this from here, Hind3 to Hind3 is the fragment they have constructed. OCT4, SOX2, KLF4, and CMYC. We also need several things for the mRNA, eukaryotic mRNA. There should be a GCAP and there should be a poly-A tail, right? So they have added a signal for poly-A tail at the end, because GCAP and poly-A tail, these are kind of circularized to increase the stability and also increase the efficiency of translation. So all the black marks they have put here, they are self-cleaving peptides. They have CAG. CAG is a promoter that is important. That is a highly highly expressing promoter. And from here till here, that's the piggyback. This whole thing is the piggyback vector. And ampicillin is for manipulation when we do within E. coli. When you are going to do your assignments, the paper presentations, I want you to go to the paper and describe the construction of the genomic, the recombinant DNA, whichever they might have made. So you need to find a paper that has some details that you can explain. Find what is each of the component and explain it, then you would have understood a lot of it. I hope you remember delta TK, thymidine kinase, and this is for puromycin resistance. We have read that in the use of it in construction of transgenic animals, positive selection and negative selection. So here they are representing it both as a single genetic element. Here also they put a poly A signal for the mRNA, for the poly A tail of the mRNA, and here is the promoter for the expression of the selectable markers. Ampicillin is for E. coli selection during manipulations in the E. coli. So I hope you understood that the general genetic construction of this vector, and that is why it is important how you engineer stuff. That will give you a much broader perspective of what all we can do and how we should do based on the objective. Okay. Take somatic cells, put in the piggyback vector, select the iPSCs, then you ensure that they grow into iPSCs, then you analyze them, and once they became induced pluripotent stem cells, you want to ensure that you have removed the piggyback vector, and once the final cells are reached, you have to ensure that there is no integration at all. Okay. In the next class, we will finish this, and I hope you understood the background and the technical things that are required. And in the next class, please try to read the paper meanwhile. There are a few things that I would want you to have a look at it, and some of the analysis of results are also there. It's a good idea if you read. Please bear in mind that I'm not going to discuss all the results. I'm only going to discuss as much as required for us. Okay. Any questions here? If you have any questions, I'll take them.